What truly sets humans apart from the rest of the animal kingdom? Is it our intelligence, our creativity, or perhaps something even more fundamental? Many scientists believe our greatest superpower is language, the remarkable ability to communicate complex thoughts, emotions, and ideas through speech, writing, and even gestures. With language, we can share knowledge across generations, tell stories that inspire and collaborate to solve problems in ways no other animal can. It's the foundation of our societies, our cultures, and our progress as a species. But where did this extraordinary ability come from? The answer, as it turns out, is hidden deep within our DNA, within the very building blocks of life that shape who we are. For decades, scientists have searched for the genetic instructions that might have given rise to language. They've compared human DNA to that of our closest relatives, looking for clues that could explain our unique gift for communication. Imagine for a moment, what if you could take a gene from a human and place it inside a mouse? Could that mouse gain some aspect of our ability to communicate? That's exactly what a pioneering team of researchers set out to do. Their goal, to see if a single human gene could make a difference in how a mouse expresses itself. They focused on a gene called NOVA1. This gene plays a crucial role in the development and function of brain cells, and it's just a little bit different in humans compared to other primates. That tiny difference might hold the key to our unique abilities. By inserting the human version of NOVA1 into mice, the scientists hoped to see if it would change the way mice communicate with each other, perhaps even altering the very sounds they make. This experiment was a bold and imaginative attempt to rewind the clock of evolution, to peer back in time and catch a glimpse of how the roots of language might have first taken hold. The big question, could a single human gene really alter a mouse's squeak, making it more like the sounds we use to communicate? Could this be the first step toward understanding the genetic origins of language? What the researchers discovered was nothing short of astonishing. Their findings open a brand new chapter in our quest to understand what makes us human, and how a few tiny changes in our DNA might have given rise to the voices that connect us all. Language is more than just a collection of sounds or words. It's the invisible thread that connects our minds, allowing us to share thoughts, emotions, and ideas in ways that no other species can. Through language, we bridge the gap between individuals, cultures, and even generations, creating a shared understanding that forms the foundation of human society. With language, we do so much more than simply communicate. We share our dreams and aspirations, teach vital skills to the next generation, and pass down knowledge that has been accumulated over thousands of years. Language allows us to tell stories, express love, resolve conflicts, and build communities. It's the tool that lets us imagine new worlds and solve complex problems together. While many animals have ways to communicate, through sounds, gestures, or scents, no other species on Earth can exchange information with the detail, nuance, and complexity that humans can. Our conversations can span abstract concepts, future plans, and even fictional universes, setting us apart from every other creature. The secret to this extraordinary ability lies deep within us, in our DNA. Our genetic code holds the blueprint for language, shaped and refined by millions of years of evolution. Over countless generations, small changes in our genes have given rise to the unique structures in our brains and bodies that make language possible. Scientists believe that a series of small but crucial genetic changes gradually rewired our brains and vocal cords, paving the way for the development of speech. These changes didn't happen overnight. They were the result of a long evolutionary journey, with each step bringing us closer to the rich tapestry of language we know today. One gene that has captured the attention of researchers is called NOVA1. While it isn't a language gene, in the sense of controlling speech directly, NOVA1 acts more like a master conductor in the brain, orchestrating the activity of many other genes and helping to shape the neural networks that underlie our most complex abilities. NOVA1 plays a crucial role in controlling how brain cells connect and communicate with each other. This intricate web of connections is essential for higher-order thinking, memory, and of course language. Without such coordination, the brain would struggle to process the complex information required for speech and understanding. What's fascinating is that the human version of NOVA1 is just a tiny bit different from that of our closest animal relatives. Yet even this small difference could have had a massive impact, helping to unlock the full potential of our brains for language and abstract thought. By studying genes like NOVA1, scientists are slowly piecing together the puzzle of how language evolved. Each discovery brings us closer to understanding the genetic changes that set us apart from other species and made our unique form of communication possible. It turns out there isn't just one magic gene responsible for language. Instead, it's the result of many small genetic changes working together, each contributing a piece to the complex machinery that gave us our most powerful tool: the ability to communicate, collaborate, and create. By understanding these genes and their roles, we can trace the incredible journey that made us uniquely human. Language is not just something we use, it's a fundamental part of who we are, and the story of its evolution is the story of us all. How do you give a mouse a human gene? Enter CRISPR, the genetic scissors. Scientists use CRISPR to remove the mouse's NOVA1 gene and replace it with the human version. The result, mice that were normal, except their brains now use the human NOVA1 gene. The difference between mouse and human NOVA1? Just one amino acid. Could such a tiny change really matter? That's what the experiment set out to test. By creating mice identical except for this one gene, scientists could pinpoint its effects. This precision is what makes modern genetics so powerful. The stage was set. Would the human gene change the way mice communicate? The scientific world waited for the answer. What happened next surprised everyone. After the genetic swap, scientists gathered around the mouse enclosures, eager to observe any changes. They listened closely, using sensitive microphones and specialized equipment to capture even the faintest sounds. The anticipation in the lab was palpable. No one knew exactly what to expect from these genetically altered mice. Normally, baby mice produce a chaotic chorus of high-pitched squeaks and squeals. These sounds are their way of calling out to their mothers, signaling hunger, discomfort, or simply seeking attention. The calls are usually unpredictable, with a wide range of pitches and patterns, reflecting the immature state of their vocal control. But when the scientists listened to the humanized mice, they noticed something remarkable. The calls were noticeably lower in pitch and much more consistent. Instead of the usual jumble of sounds, the vocalizations were simpler, more uniform, and less varied. The difference was clear, both to the ear and in the visual patterns on the audio spectrograms. All of this was the result of a single, tiny genetic change. By swapping in the human version of the NOVA1 gene, the researchers had fundamentally altered the way these mice communicated. The team didn't stop with the babies. They also recorded adult male mice, who are known for singing complex ultrasonic songs to attract females during courtship. These songs are usually intricate, with lots of variation and improvisation. Yet the humanized adult mice sang songs that were much simpler and more predictable. The melodies lacked the usual complexity, instead following repetitive and regular patterns. The researchers could see this clearly in the audio analysis software, which showed a stark contrast between the complex calls of normal mice and the streamlined songs of the gene-edited ones. Surprisingly, the human NOVA1 gene didn't make the mice's sounds more elaborate or expressive. Instead, it made their vocalizations more controlled, as if their brains were better at regulating the muscles involved in producing sound. This suggested that the gene was influencing the neural pathways between the brain and the larynx, the organ responsible for producing vocal sounds. The change seemed to give the mice more precise control, but at the cost of vocal variety. In other words, a single human gene had a real, measurable effect on how mice communicate with each other. The implications were profound, hinting at the deep connections between genetics and the evolution of communication. The experiment was considered a success, but the results took everyone by surprise. Instead of making the mice's calls more complex, as some had predicted, the gene brought a new level of consistency and control. The audiographs and spectrograms told the story. The humanized mice produced calls that were strikingly uniform, with regular patterns that stood out from the chaotic calls of their unaltered peers. This unexpected finding challenged long-held assumptions about how language and communication might have evolved in humans. Instead of complexity, it was consistency and control that emerged from this genetic change, opening up new questions about the origins of speech and the role of our genes in shaping how we communicate. The results shocked the scientists. They expected subtle changes, not a dramatic shift in the mice's calls. Surprisingly, the human gene made the mice's vocalizations simpler, not more complex. Why would a gene linked to language reduce complexity? The new hypothesis: Maybe the first step toward language was gaining control, not complexity. Like learning an instrument, you need control before you can play music. The human Nova1 gene may have helped our ancestors produce consistent, repeatable sounds. This control could have laid the groundwork for more complex speech later. The evolution of language might have started with simplification and precision. This discovery forced scientists to rethink how language began. Sometimes less is more. This experiment offers a rare glimpse into our evolutionary past. Fossils show us bones, but genes reveal how our brains changed. The Nova1 mice support the idea that precise vocal control came before complex speech. Consistent sounds may have been a crucial adaptation for early humans. The human Nova1 gene likely strengthened the brain's control over the larynx. This small genetic tweak could have set the stage for vocal learning. Evolution happens through tiny changes, each building on the last. Nova1 isn't the language gene, but it's a key part of the story. It's one of the tweaks that nudged us toward language and everything that followed. Another famous language gene is FOXP2, discovered in a family with speech difficulties. FOXP2 is crucial for learning the complex movements needed for speech. Like Nova1, scientists put the human FOXP2 gene into mice and saw changes in brain circuits. FOXP2 helps with learning movement patterns. Nova1 seems to control the vocal instrument itself. Think of FOXP2 as teaching you the dance steps, and Nova1 as helping you hit the right notes. Both are essential, but neither works alone. Language is built from many genes, each playing a different role. FOXP2 and Nova1 are just two stars in a much larger cast. The evolution of language is a story of many genes working together. No single gene makes us talk. Language is a team effort. Genes like Nova1 and FOXP2 are important, but they're just part of a complex machine. Our ability to speak involves brains, ears, lungs, and vocal cords all working together. Genes are the ingredients, but environment is the recipe. Even with the right genes, we need to hear language to learn it. Our DNA gives us the potential. Our culture and experiences shape how we use it. Understanding this complexity is key to appreciating human language. The Nova1 discovery is an exciting clue, but the full story is much bigger. The quest to understand language will keep scientists busy for years. Each discovery brings us closer to unraveling the epic story of how we learn to speak. The Nova1 discovery is just the beginning. Next, scientists will test the gene in other animals, like songbirds, to see if it affects vocal learning. This research could also help us understand and treat speech disorders in humans. By pinpointing key genes, we can develop better diagnoses and therapies. Studying language genes helps us understand what makes us human. Every new discovery is another piece of our origin story. Science starts with curiosity and leads to surprising answers, and even more questions. The journey to understand our language superpower is far from over, but each step brings us closer to the truth.